order to create a pedigree, one must take the alleles of both the husband and the wife and cross them in order to find the chances of what their children will have. For example, with this girl, Yvonne, and her husband, she has the genotype homozygous recessive, which is RR, meaning she has Velcro, while her husband has the genotype heterozygous dominant, which is uppercase R, lowercase r. So in order to find the chances of her children having Velcro or what their genotypes are, we must cross these two and find the chances, being this box. So what you do is you split the alleles into their own separate box. You put one genotype from either the husband or the wife, in this case it would be the husband, on top, and then the next person on the side. Then what you do is you cross whatever letter is in each box with the opposite box that it represents. So for example, you would cross this big R with the little r over here in order to get heterozygous dominant. You would cross this big R down with this little r in order to get homozygous dominant again. Then you would do the same thing except for this R. You would cross it with that allele in order to get homozygous recessive, and you would cross it with this allele in order to get another homozygous recessive, meaning that these two children would have Velcro. The odds, after you find each genotype, you can predict the odds in which their children will either be heterozygous dominant, homozygous dominant, or homozygous recessive, meaning they have the disease that you are trying to cross. So in this case, Yvonne and her husband would have a 50% chance of their child being homozygous recessive, meaning they would have Velcro, while they also have a 50% chance that their child will be a carrier of the disease but will be normal and will not express any of the symptoms of Velcro. But due to the situation, because we do not know what her husband's actual genotype is, we only know that he's normal, we have to assume the other allele, which is homozygous dominant. So once we do that, we would set up the Punnett square in the same fashion, except this time he has two big R's because he's homozygous dominant. So we would have those two on top, and then we would cross them with Yvonne again, who is recessive, who has Velcro, and we would find that there's a 100% chance out of this entire Punnett square that her children will not have the disease. However, they will be carriers of the disease, meaning their genotype would be heterozygous, meaning one big R, one little r, one big R, one little r, one big R, one little r, one big R, one little r. So in order to cross these two, you would have to, again, connect the big R, or the allele that you have, with the allele on the other side in order to create 4 genotypes and predict the odds/chances of their children either having the disease or being normal.